Please join me in welcoming Professor Toby Coates.
Thanks, Fiona. One strategy is to have a long introduction so you have less time to talk. So I'm going to have to speak to my PA about sending that one out again. It's nice to be among friends in the type 1 diabetes community, and I thank you all for coming out tonight, and I thank you for your support for JDRF and also for diabetes research, because without your support we can't attempt to do the things that we're going to try to do and the things that I want to show you today. So thank you from the bottom of my heart at the start. So I don't really need to talk to you about really how the impact of diabetes is on our community. And as you know, we're really focusing on type 1 today, and we have at least 140,000 Australians with type 1 diabetes. And the incidence, as you'll hear from Jenny Cooper's work when she speaks later on, is rising, and it's rising amongst our children, which is really quite disturbing, I think for all of us. We also have a very large population of type 2 patients with type 2 diabetes, many of whom the techniques that I'm going to talk about today I think ultimately will be beneficial for as well. And obviously we have people with gestational diabetes who are set up or predisposed potentially to go on and get diabetes later in life. So we're talking about the pancreas, and we're talking about a specific attack on the insulin secreting cells, or the beta cells, that make up about 1% of the human pancreas. So 99% of the pancreas doesn't really matter from my point of view, but the 1% that makes insulin is obviously critical for people with type 1 diabetes. So I apologise for these slides, but I've had them for a long time and I use them a lot. But just to remind you, that's what insulin does. It enables you to take sugar and put it into the cells. It's a hormone that essentially enables normal metabolism to happen. And when you have type 1 diabetes, of course, you haven't got any insulin, so you're not able to put glucose inside your cell. Now, type 2 diabetes, and this again is a bit of a simplification, but type 2 diabetes, the problem's not that you haven't got insulin, but usually that you've got a degree of resistance. The cells are resistant to the insulin, and so the glucose can't get inside the cells until later on in the course of the disease when the pancreas fails, and then obviously they are deficient of insulin and you need to have insulin therapy at that point in time. So this is a nice slide. This looks at what the islet actually looks like with staining for the insulin positive cells here in red and the glucagon positive cells here in green. So those two cells within the pancreatic islet provide a counter-regulatory balance that enables us to keep tight blood sugar. So one, insulin takes glucose into the cells, and glucagon helps us to mobilise sugar from tissue stores to prevent the sugars getting too low. So you really need both of them for normal endocrine function. So type 1, as I said, estimated to affect up to 140,000 Australians. And as an immunologist, it's very interesting because it's a specific immune attack only on one cell. That's quite uncommon. And as I say, the incidence is rising, and it's one of the major causes of non-traumatic death in young Australians. So therefore, it's an NHMRC priority for us to do research in it. I like this slide as well. This is from the Kids in the House, and there may be people who participated in the Kids in the House program. It's a very clever program that the JDRF run, where they make sure that we have young people with type 1 diabetes flown in to Canberra to the electorates of all of the sitting members, usually around lunchtime, so that when they give them something to eat, that the politicians have to watch them give needles and injections and things like that. And that, of course, is a very positive way to remind people in power how difficult this is to actually manage. But what I like about this slide, and I was there when they did this, was just the visual impact of 15,000 needles to treat one child for 10 years, and that was put in the lawns of Parliament House, which I think is a very strong statement and message. And for me as a researcher, it's one of the motivating factors to try to do something with the transplant that might help us get over these sort of issues. So it is an immune reaction to the beta cells, and what you're seeing here is a bit hard to make it out, but that's an islet, and here are the cells of the immune system, the T cells that are coming in and destroying it. And of course we get over this problem by using insulin pumps and giving insulin bolus injections. Okay. So insulin's 95 years old, and it's still an absolutely fantastic treatment and a fantastic medical miracle, really. And the next time you're bagging an orthopaedic surgeon, which some of us do when they're driving around in their Porsches and that sort of thing, we have to remember that insulin was actually discovered by an orthopaedic surgeon. This is Banting over here, who was an orthopod who went to work with a metabolic physician, and in the remarkable period of 8 months, isolated insulin and then was able to purify enough to treat Leonard Thompson, who's here in the middle, successfully. 8 months from bench to bedside. Now, as Nick Petrovsky and I will tell you, that's extraordinarily fast, and it's not the sort of thing that would ever happen today, because there is so much regulation, there are so many barriers that we have to actually moving things quickly. But back in the 1920s, this is what was able to happen. And in just the year after they did this, Banting and MacLeod were awarded the Nobel Prize, which again is probably the fastest Nobel Prize that's ever been awarded as well. So it is a triumph, and we're all regarded as the mainstay of what we need to treat people. But the problem with type 1 diabetes, and I apologise for this slide, I don't wish to point out the very bad side of type 1 diabetes, but I think it is important for us to notice that all of the complications, despite insulin, are significantly increased in the type 1 diabetic population. So renal failure, which is my other job, blindness, stroke, heart attacks, and in particular this nasty short-term, this very debilitating complication of hypoglycemic unawareness, which is where the blood sugars can drop precipitously without people recognising that the sugar's dropping, are extremely difficult complications to manage. And it's for this very difficult group of patients that the islet transplant therapy that I'm going to talk about has really been derived. So it is a multi-system disease. It affects the kidney, the brain, the eyes, as we heard out from Fiona at the beginning, and the blood vessels and the heart, and overall potentially shortens life by about 15 years. So the obvious question is, well, we know that the beta cells are destroyed by the autoimmune process, so why don't we replace the cells that are missing? That sort of makes sense, or makes sense to me as a transplant person anyway. So for that reason, I guess islet cell transplantation has been proposed. And this is really, I guess, if you like, a surgical type of treatment for diabetes. And you can transplant the whole pancreas, which is what Richard Lillehei did first in Minnesota in 1967, or more successfully later, Paul Lacey, islet transplantation, first human islet transplantation in 1990. But this actually isn't a new idea. This is the British Medical Journal from 1894. And in 1894, and I've just underlined it because I think it's rather cute, this was a physician who'd done some experiments where he'd extracted some extracts from the pancreas of some sheep and was trying to treat a patient with type 1 diabetes. He writes, as reporting the failure of this particular treatment, he says, especially as the possibility of treating pancreatic diabetes by means of grafts to the pancreas is likely to receive further attention in the immediate future. That's 1894. So it took a little while for 1894 to speed up. And I actually found a picture of him. I love the internet, you can find all sorts of things on the internet. So I typed his name in, thinking, oh yes, there'd be a website somewhere that had a picture of him as a hero in diabetes. No, I'm afraid not. One of these other chaps played for a Cardiff rugby club and was very, very famous. And so he's on the internet, and then just by pure accident, over there is P. Watson Williams carrying his hat. So, islet transplantation. The idea behind it is to take the islets from a deceased donor, in this particular case, isolate the cells, and then give the cells back into the liver of the recipient over here. And that's essentially the graphic that you see to explain it all. And it's a process that's accelerating over the world. And what's really interesting is that probably the leading centres in the world now are in Europe rather than North America, because there's been issues regarding reimbursement of islet transplantation in North America that still haven't been completely ironed out. But in Europe, a large number of transplants are occurring. We have no idea how many are occurring in China, but I'm sure that they're there, and we just don't have accurate figures on them. And in Australia, our activity is significantly increasing as well through the transplant program. This is an old slide, and the numbers are not really quite up to date. This is what pancreatic islets look like. This is a beautiful prep that we transplanted in December of 2015. This is insulin-positive tissue staining up nice pink colour here. This is the exocrine tissue, that's the digestive enzymes around the outside. So this is a very pure prep for somebody who's going to get an infusion of islets. So this program began in 2006, so it's 10 years old this year. We had our 10-year anniversary. And there's Tony Abbott when he was the health minister. And he helped assist get the money, and that's Susan Alberti over here who put down $5 million on the table in front of John Howard and said, this is my commitment to type 1 diabetes, where's yours? And at that point he came up with the rest of the money to start this program off. So we can really, it's a good example of how one person can actually make a huge difference. One very wealthy person, but one person can still make a huge difference in these things. So it was a $20 million 6-year grant in 2005-2006. There are 2 isolation centres in New South Wales and Victoria and 3 transplant centres. And we're in because we have a very high organ donor rate and a long-term interest in transplantation. So therefore, we've felt to be the appropriate sort of people to participate. This is what it currently looks like to do one of these procedures. So this is one we did just recently. And what I just want to draw your attention to without going too much into detail is these are very labour-intensive procedures as we currently do them, and they're done in an operating theatre with a lot of people hanging around the outside, and that drastically increases the cost of these things. And what I want to show you at the end is the new research that Fiona mentioned before that we've just been funded for JDRF International for, to try to develop different ways of actually doing this. So this is just, and I apologise to those who've seen this before, but this is our first patient that we treated. So this lady had type 1 diabetes for 36 years, had full house complications, mainly in the eyes, not in the kidneys, but in the eyes with this severe hypoglycemic unawareness. And I'll just take you through this, how this works on the sort of y-axis over here. You can see before her transplant, she has quite chaotic blood sugars going down between about 2 and 3 and up to about 20. So generally what we would all regard as not terribly good control, and her HbA1c is 9.4 because she's running her sugars high to avoid the hypoglycemia, which is all logical and makes good sense. You'll see in the graph below this is C-peptide. Now when you have functioning beta cells on a production line produce C-peptide at the same time as they make insulin. So we can use that as a means of telling whether or not somebody's pancreas is working. You can see before we transplanted there's no C-peptide because she's got no functioning beta cells. And down the bottom here is her insulin, exogenous insulin dose that's been given by her endocrinologist. You can see with the transplant, first transplant, we do see immediately an improvement in her blood sugar control, quite substantially compared to the pre-transplant. But unfortunately, and you can see, we start to see C-peptide being produced. So for the first time in 36 years, she's got her own endogenous insulin production material on board, and that's terrific. You can see though by about the 3-month mark, that's starting to wear off a bit, and there's not as much there as we want to see. So we gave her a second transplant of cells over here, and again you can see improvement in blood sugar, but an augmentation in C-peptide production over here, a diminution. We were able to wind back her exogenous insulin. Here at about day 230, she comes off her insulin, and she's been insulin independent from this transplant for the last 6 years and is still insulin independent. So it's really been a life-changing transplant for her, and obviously a major motivation for all of us. So around the country now, we have of the order of 30 people, so it's still very small numbers, but we've of the order of 35 people who've been treated with these techniques and are doing very, very well. That led, as Fiona said, to this nationally funded centre. It's the only one that South Australia's got, and I'm very pleased about it because, again, it's an indication that we can do very high quality work here in South Australia, and it's not all done in New South Wales and Victoria and Queensland, and that's, I think, important. So what about making new insulin-secreting cells? So people always ask me about stem cells, and so I'm going to talk to you about what they can do and what they can't do, and where we're up to in 2016 with them. So if we think about cells for cell therapy, we have to think about what type of cells we've got that we can potentially use. Now, obviously, there are embryonic stem cells. We're all derived from embryonic stem cells. We start off as an embryonic stem cell, and then we differentiate to become the people that we are. So that's a possibility. You could use those cells to potentially generate new insulin-secreting tissue. But as I'm sure you're all aware, that's there are a lot of ethical issues around the outside of that, the source of those cells, and so generally that sort of research is not really taken off, I have to say. So if we think about what we could use, Adult cells would obviously be a good source of cellular material because no one's going to argue about that. Autologous meaning from the same person that has the disease. So embryonic stem cells we could use, adult stem cells we could use, and we could potentially use cells from other animals. And I'll talk a little bit about that towards the end. So one concept that you will hear about is this idea of the induced pluripotent stem cell. I don't want to spend a lot of time talking about this because this is not a technology that's yet in prime time, but it's very interesting and it's sort of helping us to push the field along. Basically, the idea with the iPS cell is that we take an adult skin cell, usually a skin cell, and we differentiate it back. We treat it with a number of chemicals to make it become more immature or more primitive. And once we've done that, pushed it back to become more like a stem cell, we can then push it forward it to become something that we might want it to be. In this particular case, an insulin-secreting cell. So the iPS technology is out there and it's something that's being developed. I'm not sure if it will necessarily give us the source of insulin-secreting tissue that we want, but it's certainly going to be one of the tools in the researcher's armamentarium, and it's, I think, a very interesting technology. But what there are out there that are very, very interesting now is there are at least 3 protocols that take a sort of half embryonic stem cell and differentiate them forward to become insulin-secreting tissue. So there's at least 3 of them out there. Now, if there'd only been one, I'd be quite cynical about it and say, well, you know, one person can do something. But if you've actually got 3 separate protocols that have been developed, that usually tells you that there's something out there that's starting to look like it might be quite good. So I won't go over this in any great detail, but this is a paper that was published in Cell in 2014 from Doug Milton's group. And he is one of the groups that has got this protocol working now where they can get what looks like an insulin-secreting tissue from a stem cell. This is Timothy Kiefer's paper, which was in Nature Biotechnology. It's probably a better protocol. It's a 7-step protocol as opposed to a 5-step protocol. And you'll have to, again, take my word for it, but all of these markers that they're able to induce with this complex protocol are all the things that we would expect to see inside a normal beta cell, what's inside of you that's making insulin. So I think these are actually very, very promising techniques. And I'll just show you a few quick pictures here. And again, I won't go through these in any great detail, but these are normal beta cells in the middle here in this flow cytometry profile. And these are the stem cell-derived beta cells. So just without even going into the details, you can see that the populations of cells sort of there and there and there and there are actually very similar in terms of what they look like on these 2 graphs. There's a few more cells over here in the stem cell-derived beta cells that probably aren't quite exactly the same as what you'd see in a normal human. But certainly that population there, if you could isolate that, looks very, very similar to what you've got over here in the normal human situation. So it's very promising. And if you look ultrastructurally with electron microscopy, you can see that these are granules inside these cells that they've grown in vitro. And you can see here with the stem cell-derived beta cell, these are granules for insulin and glucagon. So these cells that have been derived in vitro from stem cells are starting to make the right-looking subcellular machinery or tools or products that we actually want. So I think it's actually very, very, very promising. Now, this is, again, a complex slide, and all I really want you to do is look for patterns here. Now, these here are the primary beta cells. This is what's normally in your pancreas when it's working normally, and these are the stem cell-derived beta cells on the left over here. So this gene map, all I want to show you here is it's similar, but it's not exactly the same. So what that's telling us is that the protocols that are out there at the moment are getting close to producing something that looks very similar to what the normal situation is, but it's not quite perfect. And I show that with this cheeky cartoon like this, and the caption, if you can't read it, says, "It's time we face reality, my friends, we're not exactly rocket scientists." So what that's telling you is these cells here, they look like it, but they're not quite exactly where we want them to be. But they're much closer than it's ever been before. So I think we're heading in the right direction. I hate giving false hope to people. I really hate that. And I don't want to give false hope to you. But what I will tell you is that the protocols that are out there at the moment, if we had a 68 kilogram human, we need between 350 and 750 million islets to transplant. We can grow 300 million of those cells that I just showed you in one 500 ml flask. In one 500 ml flask. So you need 2 flasks potentially if we can get these protocols right. So I genuinely think this is something that's coming and will come very close very, very soon. What about pigs? Okay, we've used porcine insulin to treat diabetes for many, many years before we had recombinant human insulin. These are the Auckland Island pigs, which, believe it or not, were dropped off on the Auckland Islands by sailing ships in the late or the early 1800s. And the reason that they did that was they thought, hang on, we might get shipwrecked down here. What happens if we're shipwrecked down near the South Pole and we stumble across these islands, we've got nothing to eat? So we'll drop off some pigs and we'll just leave them there. So they were dropped off and they were forgotten. And so they are actually very pure pigs that hadn't been exposed to all the viruses that commercial pigs do have now. So they're quite a remarkable resource. And in fact, what you can't really tell from this, because I don't know if any of you are experts on pigs, I'm becoming one, by the way, but this pig's actually evolved in the 200 years that it's been on the Auckland Islands, because it's very inhospitable. So to get at the oysters and the molluscs around the shore here, they've got to have extra long snouts to be able to get down there and get them. So these have got longer snouts than normal pigs, apparently. Anyway. I think beauty's in the eye of the beholder on these things, but anyway. So there have been trials that have been done in New Zealand looking at these islets, encapsulated islets, on a very small scale, and possibly showing some benefit, I think, not in terms of efficacy, but in terms of no transmission of infection, and that's really important. So I think that's been helpful from that point of view. But what really is exciting is work that's coming out of Korea, Australia, and the USA. I'm not involved in this, but collaborators of mine in Melbourne are, and that's these genetically modified pigs that have had genes knocked out for complement in this thing called GAL, which is a sugar. And we've been able to achieve now over 350 days, that's over a year of insulin independence, okay, by transplanting these cells into primates. Quite remarkable. So not being rejected. Now that still relies on immunosuppression, but it's looking very, very positive. And probably, watch this space. That's the sort of technology that's coming in. Australia's leading in it. So what about different physical barriers? The patient I showed you before had to take lifelong immunosuppression to enable those islet cells to work. So what about actually putting physical barriers to stop that in? Well, the one that you will have heard about is this. It's from ViaCyte. Now, ViaCyte have created this device here where they've taken one of those stem cell lines that I showed you before and put it inside this box and then transplanted these into, I think it's 50 type 1 diabetic patients in the United States. Phase 1 trial supported by the JDRF. And that's what the cells look like. We've not seen any of the clinical outcomes, but I know the gossip on the street from the people that I spend my time with, is that they've had big problems with these devices in actually finding them again because it's a foreign body. And what the body does when it sees something foreign like that is it mounts a fibrotic reaction, okay, and expels it in exactly the same way as when we were running around chasing, chasing polar bears or whatever. If you've got a spear in your side and the spear broke off, your body forms a fibrotic reaction to cope with that. So it's no surprise to me that if you put a foreign body in somebody, that's going to form fibrosis. Now that's important because what I want to show you with Professor Greenwood in a minute, we think may be a way around the outside of that. The second thing that's really interesting, and this paper comes from the Germans, who don't have a big background in diabetes, but they have a big background in engineering, because that's where Mercedes-Benz and BMW come from. So and that's highly relevant for this particular patient because he was an engineer with type 1 diabetes. And so he said to his physician, well, I think I can make a device that would protect islets from my immune system, and I'd be willing to have it implanted. So that's exactly what he did. So he created this device here, which I'll show you a picture of, in which there are pores and the cells are in the middle and the insulin can secrete out on either side. And he had it transplanted into himself. Now, this is not the most marvellous slide here to show you the results of, but the bottom line is, and there's a lot of issues with this, but the bottom line is that he was able to improve his hemoglobin A1C by having these macro-encapsulated cells in his body for up to a year, which was quite impressive. And essentially what he used to do at night is go home, connect that device up, I'll just show you, connect this device up to an oxygen port and pump oxygen in there to keep the cells alive. Innovative solution. Not sure that it's necessarily it's ready for prime time, not sure that it's going to take off, but it's pointing us in the right direction, I think, that these things are starting to happen. So the alternative sources for insulin-secreting tissue are science. Fact, not science fiction. They are coming, and they will be here soon. There are 3 companies here, ViaCyte, Betalogics, and Sema Therapeutics, all of which are spin-out companies from Harvard or from Yale that people are investing in, and Americans don't invest in things unless they think it's good. Three companies pushing in this direction, so I think it's good. So this is probably, prime time and trials are starting to happen, specifically with the ViaCyte. These guys would be next cab off the rank with the xenograftic cells from pigs. And in the middle, the IPS, I'm not so sure, but certainly it's going to be useful technology in terms of moving forward. But what I'm going to tell you now, despite the success that I showed you, is that the liver is really not the best place for us to put these beautiful, delicate cells that secrete insulin. And all of these factors that I put around the outside here, issues with the immune system, this inflammatory blood-mediated reaction, lack of blood vessels, low oxygen, high immunosuppression, increase in portal pressure, are all bad and are things that potentially will make are deleterious for islet cell function. And we know that when I transplant somebody at the Royal Adelaide, 70% of the cells that I transplant in the first 24 hours are dead. They die because the site is the wrong site. So you don't need to be a mathematician to know that if we could find a better site for them and we didn't lose 70% of those cells, well, A, we would leave less islet cells to do the job and we might actually be able to treat more people. So that's the rationale for the research that we've been undertaking. So the liver's a bad place because the oxygen is low. These cells really need to be connected to blood vessels. They've got high levels of this nasty endotoxin, which is a bacterial sort of product. There's the immune system. We have this problem of the islets are really thrombogenic. What I mean by that is you put them anywhere and they form little clots straight away, and they form a clot, they die. So we have to anticoagulate people to stop this from happening. So it's a big deal. We can't monitor them and we can't take them out. Now I put those in red because all the treatments that I've told you about in the last 5 minutes or so are all very, very new. Now if it was me or it was my child, I would not want something that was new, that had been manipulated by some guys like me in the lab, injected into my child. I wouldn't want that. I would want to be able to get those cells out, and I want to be able to look at them in case they turn nasty. Because whenever you manipulate a cell, if you manipulate a cell and push it, make it do stuff, it potentially becomes cancerous. So to me, it's a no-brainer that the liver is not the right site for any of these new technologies going forward. So with that in mind, what we need to do is look for alternative sites for these new cells. And these are the sites that people have talked about, such as up in there, that's actually the thymus, although they've actually drawn the thyroid. I think Nick will agree with me there, but because the thymus should actually be down here. It's a Japanese paper, I apologise for that. But anyway, the thymus into the muscle, the liver obviously you know about, the spleen's been used before, the testis, that's going to rule out half the population already, but people have talked about that and I'm not so sure you'll have many men lining up for that to happen, but I'm only saying what people are talking about. The bone marrow as well, but the site that I'm really interested in is under the skin. Because if we could put them under the skin, that would be minimally invasive compared to putting them in the liver. Technically easy. We'd have the option to remove the graft if we had to, and we have the option to image it, and I'll show you pictures of how we do that in a minute. The disadvantage of the subcutaneous site, and it has been tried for many, many years because it's obvious, is there's not enough blood vessels. So what we actually need is a solution that potentially gives us more blood vessels under the skin that might enable us to put our delicate cells in that spot. And that's where this fellow comes in. So this is John Greenwood. He is a pioneer burn surgeon, and he was South Australian of the Year this year. He's just handed it over to the new South Australian of the Year, and he was obviously one of the runners-up for Australian of the Year. And he got his Order of Australia for developing, or for basically treating the Bali bombing people, and did absolutely remarkable work there. And his mission has been to try and eliminate the skin graft. That's his focus in life. And what he's done over the last 10 years is develop this, which is a polyurethane scaffold that he uses for treating wounds, and it's called the BTM. BTM is the abbreviation that you'll hear me talk about for the next 5 minutes or so before I finish. So this is it's a polyurethane. It's fully biodegradable. He spent 10 years developing it. It's bonded to an epidermal seal, and I'll explain why that's important in a minute. He's done safety trials in large animals and pigs. It's ISO 10993 accredited. He's got at least 2 ongoing trials. He has 3-year human safety data. We've got a licence to use it at the Royal Adelaide, so it's TGA approved. It's been FDA approved, so it's passed that regulatory hurdle, and it's FDA 510K approved as a low-risk device. So it's been through a lot of those hurdles that you might actually need. What does it actually do? Apology for the gross slide. So this is the sort of burn, or this sort of wound that he can actually treat. This is a donor free flight SAP, where nowadays, if you have a nasty cancer or something and have to reconstruct your face, what they'll do is they'll take a pedicle, they'll cut a bit of skin out of here with a blood vessel, which creates a wound like that, and then they'll transplant that up onto your face. Very, very clever stuff. The problem with that is it creates a wound, and you can see the wound there like that. This is the BTM, which has been transplanted or grafted in there, and it's polyurethane foam, and it's stitched in on the side. And what's remarkable about it is by day 5, see how red it is? So just before that was pure polyurethane, and in 5 days, blood vessels have started to grow up through into this remarkable material. By day 15, it's looking much better, and there it is by day 22. And here at day 36, when he pulls that seal off that I told you about, it's got a bit of polyurethane sticking on the top of it, okay? And that's important because polyurethane, what that actually does is it tells the body not to contract, because it tells the body that this wound is already healed, because you're not losing any water through it. That's what's so clever about this. But when you pull that seal off at day 36, look at all those beautiful blood vessels that are growing under the skin there. And then he can go on and put a skin graft on the top of it like that. And by day 75 over here, it's almost perfectly healed. So quite remarkable. So this is what John's been doing. Now, that's all very well. I'm not really interested in burns. I'm interested in diabetes. But when I saw this, he showed me this picture, hunted me down and showed me this picture. These are endothelial cells, which are blood vessel cells that are growing around the outside of those fibres that John's put into people. So this is a person. This is real. This is not a mouse. And these are human blood vessels growing around the outside of it. And as it so happens, the diameter of these cells is of these fibres is 150 microns, which is exactly the right size for an islet. So when I saw this, sometimes you get these, well, occasionally you get these eureka moments where you go, oh my God, I know what that is. That's what I've been looking for the last 10 years in islet transplantation. It's a vascularised alternative site under the skin, potentially. And it's purely, if you like, a sort of serendipitous thing, because he's a burn surgeon who works on level 4 at the Royal Adelaide, and I'm a kidney transplant guy who works on level 9, and we don't, you never see these people, so they wouldn't, just he happened to hear about what I was doing and came along and said, would you be interested in this? Because I've thought for a long time that my matrix might be good for transplanting islets. So here's that day 36 again. What I just want to show you here is there's that epidermal seal on the top over there. And what you see here at day 23 is this beautiful vascular space forming underneath the skin in somebody, and that's a human, that is not an animal model, that's in a person. And we can image this. So this is an in vivo scope, and in vivo scope is a very expensive microscope that enables us to image down to about 2 millimetres into the skin. So unlike what we're doing in the liver, what you're seeing here, these little squirrely things that are shooting around over here, okay? These are blood vessels, okay? So you can actually see the blood moving under the skin using this technique. That's the BTM over here like that. So we've got a way now where we might potentially, using this technology, image what we're going to transplant under the skin. Just to show you here, this is normal human skin, not a lot of blood vessels. This is human skin that's had the BTM in it, and you can see the blood vessels. And this is 228 days after engraftment. So, of course, I'm sorry, we have to talk about mice, because I'm a researcher. So we've tried this out in mice. So this is some BTM. We've engrafted some mouse islets under the skin here, and you can see a nice viable mouse islet there. Just in higher power, and you can probably see a blood vessel growing in around the outside over there, so that works. This is fluorescence microscopy. This is the BTM over here. Islets are showing up here nicely in pink, again the same stain that we use to identify insulin. And around the outside of the material here you can see a blood vessel growing in. So again, this is experimental proof that at least in small animals, islets will survive within this matrix. We can take them out and we can expose them to low and high glucose, and there is no impediment of a function for them being in it. So the islet, the material itself is not toxic to the islets, and that's actually very important too. And this is just small pictures showing this. Green is good and red is bad. So there are lots of happy green cells here, and this is a cell actually inside the BTM looking pretty happy. So the ultimate proof for us is to then take these cells and do them in mouse model transplants. So we've engrafted this under the kidney capsule here. This is the BTM with islets in it. It's a bit hard to see. K for kidney over here. This is standard how we do mouse transplants to show that things work. You can see the diabetic mice are immediately cured when we engraft them with the BTM that's got islet cells in it. So how would this look for you? Or how would this look for the future? What are we going? Why has the JDRF decided to give us some money to try and do this? Well, the idea would be to create a very small wound, so nothing like the wounds that are needed to treat burns victims, to then insert the matrix in there with its epidermal polyurethane seal over the top, pull that off after about 30 days into what we've already created, which will be a nicely vascularized subcutaneous matrix, and then seed the islet cells that we'd want to transplant in there, and then ultimately close that up with a primary suture. So the idea that we have is that we would take it from this, which is a very large clinical team, 5 to 7 days, very invasive procedure, to this, something that we would do into the upper arm or potentially into the abdomen with a small elliptical incision that would give us the ability to image the cells as I've shown you, and also the ability to actually take that out. And that could be taken out by the most junior surgical resident that you could possibly need. So we hopefully take this from being an inpatient procedure, a dangerous inpatient expensive procedure, to a much less invasive outpatient procedure, which we hope, if it all works out, will improve things. But why I think this sort of technology is interesting and important is it would be suitable for all types of cells. So we could use it for allogeneic, autologous, we could use it for stem cell derived islets. It's retrievable, it's modifiable. Now, I didn't have enough time to talk to you about this because I could talk for hours and hours and hours and hours, as you probably worked out, but we can actually modify this material so that it secretes immunosuppressive drugs. We've done that. So we know, so we could potentially create a little pocket that suppresses itself. So that's one of the things that one of my students has done in the lab. Not only that, but we discovered the other day, believe it or not, that bacteria and the material itself actually seems to kill T cells. So the cells that cause the rejection, when they come into contact with the polyurethane, die. So we're chasing that down as well because, hey, if you could do it without any immunosuppression, that'd be even better. But, early days and research is research. Sometimes you go down blind alleys. I don't know. So I think we've got a competitive and safe advantage in man for this sort of technology. So in conclusion, I hope I've shown you that islet transplantation is a useful treatment for selected patients with type 1. I really do think these alternative sources of insulin-secreting tissue are very close, and they're coming. So I'm not holding out false hope, but I do think that's where the field is heading. And if we can come up with some alternative sites of transplantation, it's not just me, there's all sorts of other people around the world doing this, that we may be able to offer those newer treatments in a safer manner that may be the bridge to the next therapy for transplantation. So I will stop there. If you're interested in following what we do, Kidney Transplant Diabetes Research Australia, you'll find us on the web, you can follow us there. Our friends, the JDRF, are fantastic at promoting what we do. We're supported by the NHMRC, we're supported by Cell Therapy Manufacturing, University of Adelaide, and the old Royal Adelaide, soon at some point, don't ask me when, the new Royal Adelaide as well. Thank you. And these are all the people I just give the talks. The people that really do the work. John Greenwood's a remarkable man whose vision for transplanting the skin is really his idea, and I'm really just facilitating it. These are people in my lab. This is the Eylet program. This is the Australian Eylet Consortium, and these are hepatobiliary surgeons who are involved in doing all of this. So I will stop and take any questions that you have.